What's wrong with this? Yeah, okay. All right, students. Yeah. So, good morning, and we come back again to the structure of dynein. And that's because in the last class I could not really finish what I wanted to say about dynein. If you recollect what we have done so far, and I would go back again to the same old slide of dynein to emphasize that we have so far seen that there is this triple AA plus domain in dynein polypeptide, and there are 6 such AAA domains which form a ring-like structure. And the most important ATPase site here is on AAA1. And what I would also like to emphasize is that this AAA1 domain is here, which is almost diametrically opposite to the point where you have the microtubule binding domain. So if this is the ring, and here you have the microtubule binding domain, then the AAA1 is here, and the microtubule binding domain is at the end of the stop of this. Okay, and what we saw the last time was simple but very elegant method to determine the step size and the walking pattern of these motor proteins such as kinesins and dyneins. So what they did, and I would like to recap this, what they did is they took a dynein and truncated the N-terminus, because the N-terminus is mostly involved in cargo selection. So you truncate that, you do not need the whole thing, but you add certain other domains to it, which would allow you to tag the dynein with a very specific fluorophore. So what did they do? They added some sort of a enzyme whose substrate is fluorescent. So that way you can really sort of vary the fluorescence that with which you can tag the dynein. So they did that, and they used certain other methods to dimerize the dynein. And this included adding GST, glutathione transferase, because GST, the protein, is known to form very tight dimers, or adding DNA stretches, because DNA is known to form double helices. So because of its tendency to form double helices, it would also dimerize the molecules to which it is attached. So in the end, what they have is these 2 dynein molecules which are dimeric. One is fluorescent red and one is fluorescent blue. And by very specifically localizing the fluorescence on the slide, they are able to measure precisely with time the position of these 2 arms. So the red arm and the blue arm, where is it positioned? That is what they have done. And they have used turf microscopy, and last time I explained why turf, because turf can image fluorophores which are at the boundary of the glass and water interface, and therefore if it can do this, it can get rid of all the noise that occurs because of the bulk solution, and you can very precisely with high resolution image the events that are occurring very close to this glass surface. So if you lay over the glass surface microtubules and then you add the dyneins, you'll be able to see dyneins walking on those microtubules. And if you now look at how at individual positions with time, this is the trace of the blue arm, one of the arms, this is the trace of the red arm. So you see the 2 arms can be together, one arm can be behind the other, then this arm can be in front, both the arms can walk together. So dynein, unlike kinesin, does not have a very regular movement, and that is also depicted here in this. What is depicted here is that this is time and this is spatial resolution. So this is the distance it moves. So let's say at time point 0, they are both together at the same spot. Then you see After some time, the red spot has moved back. So it seems the dynein has moved back. But then the dynein, this red arm, flips back to the same space. So it's moved back, come back to the same space. In the meantime, the red arm, the blue arm has moved forward. So you see this step. Then the red arm moves forward. Then the blue arm moves forward. Then the red arm moves forward. So this motion of one hand over the other, hand over hand motion, is something that kinesin does. But if you look here, you see that it's not hand in hand. It's as if the blue arm moves, the red arm simply catches up with it. The blue arm moves, the red arm catches up with it. So you can have hand, or you can have one catching up with the other, one catching up with the other. So the step size, the stepping pattern of dynein is not as regular as kinesin. Kinesin has a very regular stepping pattern of hand over hand. So if this were a kinesin track, instead of seeing this, you would see a repetition of this pattern, repetition of step-like patterns. But for dynein, you have a mixture of all kinds of patterns. What is also interesting from this is that you can see, so think of each of these grid lines as parallel protofilaments on a microtubule. And it seems that the two dyneins are on two different protofilaments first, and then they can have different ways of working on those two protofilaments. And you can see that here. Another thing to understand from this is the step size. So for kinesin motors, we know that it has a very regular step size of 8 nanometers, which is the distance between two adjacent alpha-beta tubulin dimers on a protofilament, right? So one step, the next step is on the next alpha-beta dimer in the protofilament. But for dynein, the step size is not uniform. As you can see here, this is one step, this is another step. But this is one step, this is another step. So step sizes are not uniform. It could be 8 nanometers, 16 nanometers. On an average, it is 16 nanometers, but it could also be more than that. So step sizes for dyneins are not uniform. And you can see the step size distribution here. This is the distribution. So whatever they have observed here, if they count the number of events, they count the number of step sizes which are 8 nanometers, 16 nanometers, and step sizes which have moved forward as in +8, +16, and step sizes which have moved backward, -8, -16, and you plot them as a histogram in terms of number of times you see those step sizes versus the step sizes, then they get this kind of a plot. So on an average, you can see the step size is about 16 nanometers, which is twice the distance that the kinesin was following. But you can see the step size can go even up to 32 nanometers and sometimes even more. And you see that a considerable number of step sizes are also taken backwards, which again indicates that the dynein motor is unlike kinesin, unlike myosin. It's almost as if so we've all watched Charlie Chaplin movies, right? I hope even if you've not watched movies, you've seen clippings. So how does he walk? He doesn't take step sizes like us. He walks like this. And then sometimes he hops. And then he walks like this. So you take that analogy and you put it on dynein, and you can think of dynein as the Charlie Chaplin of movies. Okay. Now, this I put here just for you to have a view of where exactly the dynein might be binding to microtubules. Now, although I said the step sizes are not uniform and the way it walks is also not very uniform, even though these are all true, what is also true is that the binding site on microtubules is very specific. It does not mean when I say that the step sizes are different or that it can walk forward, backward, does not mean that it can bind anywhere and everywhere on microtubule. The point of binding is very specific, and the specificity arises because of the three-dimensional conformation of this end, the microtubule binding end. And so the microtubule binding end has a specific conformation that goes and binds to the region that is between the alpha and beta tubulin. So in a dimer, in an alpha beta tubulin dimer that is part of the protofilament, the sort of groove that is formed between the alpha and beta tubulin, that is where this microtubule binding domain of dynein goes and binds. So it has a very specific binding site. So just to emphasize this, binding site is very specific. That does not change. It does not mean dynein can bind anywhere on the microtubule. It binds very specifically. What I'm trying to say is that even though it binds specifically, it can take a bigger walk, it can take a smaller walk, it can take a forward jump, it can take a backward jump. Most often, because it has a cargo attached with it, this makes the backward jump difficult for it to do. It goes in the forward direction mostly. If you remember kinesins, the single chain kinesin that we had talked about previously, you saw that when it is a single chain, it does have the propensity to go backward. But if you take a lot of these single chains of kinesin and bind to a bigger cargo, then this backward motion is inhibited and it keeps moving forward. So you can take that same analogy, put it to dynein. So this is the last slide for dynein, and I will stop talking about dynein after this. The interesting thing about dynein is that because it has, again, to go back, it has these huge number of domains and subdomains with different functions. And what dynein has done is that instead of having them in different polypeptides and combining them into a complex, so like a DNA polymerase complex or even a ribosomal complex, instead of doing something like that and combining them into a complex, it has taken up these parts, put into a single polypeptide chain. But in the end, each of these domains are going to fold independently and do their own function, just like you saw with AAA domains. So there are some other parts of dyneins that we should be also looking into. One part is this linker region, which comes right after the N-terminus. So the N-terminus is something that's going to help in dimerization, selection of cargo, right? And in modulating the properties of this dynein molecule. You have the linker that comes right after it, and the linker seems to, so if you have a donut, that's the AAA domain ring, and the linker sort of sits flat on that ring. So think of it as something that falls flat on the ring. And therefore, if you have some change in the AAA domains because of ATPase activity, otherwise, the linker position can also change. Associated with this ring is the coiled-coil-like structure, which forms the stalk that has the microtubule binding end at its tip. So it's very difficult to have a crystal structure of this entire complex. What they did was that they combined crystal structure data with data from cryo-electron microscopy to understand how the different parts, specifically the linker, the stalk that binds to the microtubule, and the associated N-terminus that is carrying the cargo are positioned as this dynein motor is walking on microtubules. So the whole thing is this: if it is walking on microtubules, then it should have various conformations, right? It should have a conformation where it binds to microtubule, where it creates the power stroke, where it perhaps detaches from the microtubules, and each of these should have a different conformation. So the idea was to understand how the ATPase activity and microtubule binding activities translate to mechanical motions within the dynein molecule itself. So to do this, they took dynein and they either had it in a conformation where it is bound to ATP or an ATP analog, or not bound to ATP, that is free of it. And then they tried to put it on microtubules, and they did cryo-electron microscopy to understand what kind of structural conformations it could have. So these images that you see are of dynein under cryo-electron microscope itself, with either ADP plus BI, so something that is going to mimic the ATP plus BI conformation, or APO, that is, there is no ADP, nothing bound to it, and then they just put it under the cryo-electron microscope and try to see how this looks. And they have to take like thousands of such images to reconstruct a possible configuration for the positioning of the linker. This is the linker here. Positioning of the linker, positioning of the coiled stock, and position of the end terminus that binds to the cargo. And you see very beautifully what they have captured here in this paper is that it shows snapshots. So if it is not bound to any ATP or ADP, you see there is a certain, so let's think of it as positioning of the linker. So this would be the linker somewhere here. This is the linker. This is the linker. The positioning of the linker, positioning of the microtubule binding domains. This is the microtubule binding domain. This is the coiled coil. So positioning of the coiled coil, positioning of the coiled coil, and positioning of the end terminus that binds to the cargo. So at each of these states, you see that the positions of these three components changes with respect to each other, and that change is brought about by whether the AAA domain is bound to ATP or ADP at all, or it is bound to ADP plus PI or ATP plus PI. So depending on this, you have a change in the orientation of these subdomains. And that shows that the hydrolysis of ATP, how it brings about conformational changes in the dynein motor, such that you can have dyneins walking on microtubules. How do you make it walk? If you have to make something walk, you have to make something move, right? Some motion has to occur. So the motion has to be translated throughout the molecule. But you have a very big polypeptide of 500 kilodalton protein. And then there is one specific region, just one specific region that is hydrolyzing ATP. How do you translate that to the entire molecule? Exactly how it is done is perhaps partly known, but what happens when when ATP hydrolyzes, or what happens when ATP binds? This is the snapshot that presents to you exactly what is happening. How the three parts are moving with respect to each other every time there is ATP hydrolysis or when there is no nucleotide bound to it. So this gives you kind of snapshots for you to literally understand. We have all seen it from the very first lecture that the way the biological motors work is that it is dependent on some sort of an energy source, and the energy source is usually ATP or GTP. And the second thing is that what this energy source does is that it changes the conformation of the proteins. And the crucial thing is to understand how the conformation of the proteins, or how much the conformation of the proteins changes. In the first lecture, I had shown you that the conformation of change in RAS. So the conformational change in RAS was perhaps just a little bit of one alpha helix moving slightly. Here you see the conformational change is quite drastic, where you see the arms moving precisely in different angles so that the entire dynein motor can function. So with this, I will stop talking. Hello, can you all hear me? I had a notice saying bad quality.
Yes, we can see. We
Yes,
can
okay,
hear.
you can see, you can hear, right? All right, all right. So with this, I stop talking about dyneins, and I will move into something else. So I stopped talking about dyneins by saying that in the very first lecture, I had mentioned that for biological motors to work, there are certain key requirements. If you think of biological motors and motors that are present in a car, or motors that run your pump at your home to supply water to your taps, to your faucets, if you compare them, there are certain key differences. One difference is definitely of size. The motors that work in a car or in a household pumps are large, they're macroscopic, right? Whereas biological motors, they have to work inside a cell, they have to be really, really tiny, really microscopic, so that You have to use very clever photonics to see them. So you are so far you've seen that you use very high definition, high resolution microscopy. You use something like TURF with very high resolution powers to actually see the spots of fluorescence as they move, or you put it under an extra crystallographic system to get the extra diffraction pattern so that you can see the structure of it, or you put it under a cryo-electron microscope to see it. Right? You cannot see it otherwise. The second difference that we had said was that they work in an aqueous environment. So the aqueous environment has a lot of limitations. Now, if you translate that to, say, the bacteria, now let's move on to something different. So you think of a car, a Formula One car that is racing on track, right? It has a certain speed. If we were to stand in front of it, we would see it just whooshing past, right? It just whoops. Now think of a bacteria. The bacteria to us, if you just would like to visualize, okay. For 1 second, I will stop sharing the screen, and what I will do is I will start sharing, and I will send you all the link of the YouTube video so that it's you all have it. Okay. Can you see this? Is the screen visible?
Yes, ma'am.
Yes, ma'am.
Okay, so these blobs are actually bacteria, and this tail is the flagella of the bacteria. So what I'm doing now is I'm playing to you the the way the bacteria is going to move. Just have a look at it. So this is a bacteria that is moving in an aqueous environment, right? Okay. So you have a bacteria that's moving in an aqueous environment, and if you so I just tried to compare a Formula One car with a bacteria. Formula One car is moving very fast. To you, the bacteria is moving slow, right? It seemed like it is moving slow. Did it seem to you that you cannot compare a Formula One with a bacteria? No, no replies. Okay. So
And but maybe the size.
yeah, yes, of course. We are talking, so you have, when we are talking about a Formula One car, we are talking of looking at it at the realistic scales. And here we are looking at micrometer order scales, right? Even the images at micrometer order scales. But the point here is size is okay, but the point here is think of a Formula One car trying to move through water. And then you will understand what is the difficulty that the bacteria is facing. The bacteria is facing the difficulty of trying to move through an aqueous environment, which means that there is a certain level of viscosity that it has to withstand when it moves. If you now take the Formula One car, put it into water, put it with the same kind of engines that would perhaps sustain in water, you will see the speeds would not be the same. Okay, so that is a key thing to understand, that the bacteria or the bacterial systems are moving in water. Despite this, their speeds are quite impressive because they can move almost 20 body lengths per second. So we cannot move 20 body lengths per second. Can we? I mean, I don't know. For 100 meters, I will be panting. I don't have the strength to run. Maybe the youngest amongst us, Ratnesh, may have the highest speed, and even he will not be able to move 20 body lengths, that is 20 times his height per second. That's an impressive thing to be done for a single cell like a bacteria, which is considered to be very primitive. So, how does it actually do this? Yeah, this is a very interesting question. How a bacteria, which is so tiny, can move quite so fast and in an environment like water, where it has to withstand very high viscous drag. So, if it is trying to move forward, the viscosity of the solution would try to inhibit it. It's almost like, so how many of you know how to swim? Nobody can swim? legs in water and try to walk in water, maybe a pond, a river, a sea. Yeah, okay. So how does it feel versus walking on land? How does it feel walking on water? Is it natural? Is it okay? Do you feel it's different? Why? What do you feel when you try to walk on water?
There is more resistance. Like in air, there
There's
is
more
no
resistance.
resistance at
Exactly,
all.
there is
Like
more resistance. You really have to spend more energy. It's like I think swimming is the best exercise because you have the water exerting something called a viscous drag. It tends to it's as if you're pushing or putting a force on it, but this force that it kind of the viscosity kind of sort of prevents your forward motion. All right, so let's now go back to. I've shared the wrong thing. Let me stop sharing this. Stop sharing. Share this. Okay, so how does the bacteria manage to do this? Bacteria manage to do this by an impressive mechanism called the flagella. And when I'm talking about flagella, students, please don't get confused because bacterial flagella is not like eukaryotic flagella or eukaryotic cilia. Because when we talk about eukaryotic flagella, that might be in the sperm or eukaryotic cilia that lines our airway passage. So when we breathe, the airway passage has these epithelial cells with cilia that keep vibrating so that the particles, if there are some particles, it can just flush it out, right? Those we have seen are made up of microtubules and dyneins, right? We've seen it so far. You will have those doublet microtubule rings, and then you have dyneins in between them, and then the dyneins walk between those microtubule protofilaments, causing sliding motion of protofilaments. That translates to motion. How this translates to, it's not very clear, but this much we kind of understand, and I think you might have also read it in your biology textbooks, 11, 12, that the sliding motion is causing movement of those appendages. Bacterial flagella is different. It doesn't have microtubules. So there are no microtubules in this flagella. Rather, there is a protein called flagellin. But it's also wrong to say there is a protein called flagellin alone because bacterial flagella is tremendously complex. It has nearly 60 proteins, 60 distinct proteins, and all of this come together to power this single appendage called flagella. Now, bacteria can have one flagella, can have two, can have multiple flagella. And also, bacteria have different sizes. So, because you're all from biology, I think it's easier for me to explain that we know that there are some things called gram-positive bacteria and gram-negative bacteria, right? And we know that the way they are distinguished is because some can retain the crystal violet stain after destaining, some cannot retain the crystal violet. Okay, how many of you know about gram staining? हाथ उठा लीजिए, बस वही काफी है. Or do I have to explain gram staining? Okay, all right. So let me explain gram staining. Thank you, Ratnesh and Sarika. So you have bacteria which are gram-positive, gram-negative, and this comes from the name of the person called Graham. And Graham, what he did was he was trying to observe bacteria at a time when nothing was known about their structure. So what was known is that you can have a microscope, a simple microscope, you add some stains, you will be able to see them. And he was trying to see bacteria in patient samples like lung tissue for, say, tuberculosis sample or some things like that. And he used different kind of stains. So he used crystal violet, and he saw that some bacteria, that crystal violet being violet, it stains bacteria beautifully well. Violet stain like that. Okay. Now, if you wash this with alcohol, some of the bacteria retain that stain, some do not. So this made a distinction and gave us the idea that there can be two kinds of bacteria. One that seems to retain stains one that seems to leach out stains after ethanol wash. So the ones that retained stains were called gram-positive. Ones that leached out stains were gram-negative. Later it was identified that this gram-positive and gram-negative bacteria are distinct because of the nature of their outer surface. So all living organisms have a plasma membrane, but not all organisms have cell wall. But bacteria have cell wall. Bacteria have something called peptidoglycan cell wall. Turns out the gram-positive has a very thick peptidoglycan cell wall, and so once it is stained, it cannot really leach out the stain. Gram-negative has a very thin peptidoglycan layer, and on top of that, it has something called an outer lipid layer on top of it. So because of this, ethanol can disturb lipid layers, and the peptidoglycan is also 1 or 2 layer thick, not too much. So the stains can be leached out. So this turned out to be a beautiful way to distinguish bacteria when nothing was known, no DNA sequence, not even what DNA was. So it was at those times. So why I said gram-positive and gram-negative is because when you think of flagella, there is something also to be which I will discuss about this gram-positive and gram-negative. So you have these two kinds of bacteria, gram-positive, gram-negative. Both of these bacteria, both of these types of bacteria can have different shapes. So you can have round bacteria, really round. Under the microscope they will look round, but of course they would be spherical, so they would be called coccus. Then you will have bacteria, if you've seen a bacteria classically represented, it's usually rod shaped, as you see here on the slide, and it's called a bacillus. Then you can have spiral shaped bacteria like spirochetes and so on. So bacteria can have different shapes. But what is crucial is that the bacteria is a single cell. And these bacteria need a way to power their movement. And the power that the way they power their movement is using this flagella, which is the motor that drives their movement. So the flagella can be present on one end of the bacteria. They can be present on two ends of the bacteria. If you think of a rod-shaped bacteria, it can be present on two ends. It can be present throughout the surface of the bacteria. So there can be various ways of positioning this motor with respect to the cell body. But all of them together would be powering the bacteria. How does the flagella power the bacteria? Before we go into that, I have a video to show it because I found it much simpler to do it rather than to keep just going on and on about this is how it moves and that is how it moves. The video will make it very evident. Okay, so this is what I was talking about. So you can have different ways by which you can position the flagella. So you could have it polar flagella, that is you can have it at one end. You can have flagella that is coming out from the sides. You can have flagella that comes out all throughout the body. And it's not important to remember the names or anything here. It's just for you to understand or have just a visual representation of how the flagella might be positioned. So if you have a motor for a car, where would you put it? Tata Nano put it in a different position compared to the rest of the industry. And if you think of a motor that is flying an aeroplane, that would be positioned in a different place, right? How many motors you would put? You would put one motor, you would put two motors, you put four motors, you put 6 cylinders, 8 cylinders. How many cylinders do you put to drive your car? So similarly, that's a question these bacteria have. How many motors do I put? Maybe one is enough for me. Some have one, some have multiple. Okay. Very interesting thing. There is something called a spirochete. And what spirochete does is it's a spiral bacteria. So it's almost like you've seen a corkscrew, something that you use to open a cork. So it is spiral in nature. So spirochetes are spiral. They do not have flagella that comes out of the body. Very interesting. They have flagella that is inside their body. So between the membrane and the periplasm, the outer peptidoglycan, there's a layer called the periplasm. And there is where the flagella lies. And the flagella, the movement of the flagella makes these spirochetes rotate. So it's like a spring which is rotating. It's pretty amazing how bacteria have evolved mechanisms for movement. Okay, So I would okay. Let's look at this first. So far we have talked very about um, how bacteria power the movement. But if you would like to understand how these motors actually look, again, what you have to do is use electron microscopy, because these are so tiny, you wouldn't be able to see the components of a flagella in light microscope. It's just not possible. In light microscope, what you can do maximum is use a fluorescent stain, use good quality imaging to see a flagella. But mostly, even to see the flagella, we go generally to electron microscope. And if you have to go into the components of a flagella, then definitely electron microscopy is the only way out, because light microscopy will simply not be able to give you the resolution, because of the, again, the wavelength uh, problem. All right. So let us uh, let us look at first this slide that we had presented in the very first lecture. And what this slide was, with this slide, what I tried to do was to show you how a motor looks. So this was the image of the motor that was designed by Tesla. And I had said that this is a microscopic system, and in our lectures we will talk about microscopic system. And I'm going to use the same slide today to emphasize that, you see, something that Tesla had designed, nature has done exactly that, and with very high efficiency. So what Tesla's motor has is something called a rotor. So this is a drum that rotates, and these are coils, induction coils, that are called, and this entire region is called the stator. So you have a component of the motor that rotates and a component that is static. If you translate that now to the bacterial flagella, you see that the bacterial flagella has exactly that. It has some components which are the stator, but the stator, just like in the Tesla's motor, which was actually driving the rotor, the stator in flagella also does the same thing. So there is a component, I'll come to exactly, with the video I'll be able to explain exactly these components to you, but for the moment let's understand that there is an analogy. So you have these induction coils, the stator, but they are producing the energy that's driving the motor, the moving component of Tesla's motor. Similarly, in flagella, you have the rotor, the flagella, that is connected at the bottom to the to the cell body. There is a component there that rotates, that is the motor. But the motor is driven by stators, which are kept in position very close to this motor, and they provide the driving energy for this motor. Exactly. So it's the exact analogy, but it's a very efficient design, highly efficient design. So for this, let us now, now move uh, to the video. I will stop sharing the screen once. Stop sharing the video. Okay, can you all see the screen? The same screen where the bacterial flagella was shown to you. Is it visible?
Yes.
Yes. Okay. Now I'm going to play this video for like 15 seconds. Just let me know if you can hear what is being spoken with the video. Okay, just give me a second. Let me increase the volume. Okay. I'm going to play the video. Just let me know if you can hear what is being said along with the video. Rotobacter cells have one flagella each, whereas E. coli cells have multiple flagella to rotate in others. Okay. Could you hear what was being said along with the video?
Yes, ma'am.
Okay, perfect. Then let us play this video once more. Okay, so we stop here and we come back. So what you heard is part of it what I have said, that you can have bacteria with various numbers of flagella, you can have various positioning of the flagella with respect to the cell body. But there is another thing that was said here is that it's a helical flagella. Helical flagella is not because you're seeing that it's going in a helix-like way. It's when it's moving, you thought that it's going like a helix, right? As a helical way, mein flagella move kar raha hai. It's not because of that. The reason they say it's a helical flagella is because of the way the flagella proteins are present inside the flagella. So the main flagella that's out of the bacteria that you can see moving in the video is made up of proteins called flagellin. There are a number of these flagellin genes, a number of these flagellins which make up the entire flagella, but the one that you're seeing in the video is made up of one single protein called flagellin. And this flagellin, when it forms, it starts depositing in a helical fashion. So how does a flagella form? It has to be produced from inside the cell, right? So as it starts building from inside the cell, you will see that in this video itself, you will see an animation where they're showing how the flagella is built. So it is built in a helical way. So the individual flagellants start depositing, rising. So it forms like a left-handed helix. It's like a left-handed helix. What does a left-handed helix mean? So the orientation, the way the helix rises is like this. So this is the way it would move, and this is the direction of rise of the helix. That's why we call it. So I hope you all now understand what left-handed and right-handed means. So right-handed would be they would start moving like this and move up. Left-handed would mean they would start moving like this and move up. So that's a left-handed helix. So they are calling it the helical flagella because of the way the flagellar proteins are going to be deposited that gives the look of a helical conformation for this flagella. That explanation, I'll go back to the video. Okay, I will stop here again. So what they said was that each flagella is 20 nanometers wide. If you think of the total body length of a bacteria, for E. coli it would be about say 2 microns, for Bacillus would be about 4 microns, so 4 micrometers. And so those are micrometer scales, and the flagella is a nanometer scale. So you have the width of about 2 nanometers. 20 nanometers. If you compare this with the step size of a kinesin, it's about 8 nanometers. Just to give you a perspective, everything here is nanometer scales. Oh. Oh yeah. So there is another information that was given here. The speed at which the flagella rotates, because it's a motor. Because it's a motor, it should have an RPM, right? Rotations per minute. So we know that the higher the RPM, the faster the motor is going to function. So your cars that are running at very high speeds would have motors with very high RPM, right? If you compare it with an airplane, maybe even higher RPM. So how much is the RPM of this motor, if you think of it in that sense? So the rotations per minute, per second, sorry, here the, एक सेकेंड मैं खुद कंफ्यूज हो गई. It's RPM, RPS क्या था? 1 second, I'm very sorry. So, sorry, it is 300 revolutions per second. So that's a very high, that if you now convert it into RPM, revolutions per minute, you can understand that it can go very, very high. So rotations per minute for a bacterial flagella is very high compared to a macroscopic motor such as the motors that we see in our car engine or in an airplane engine. That means that the efficiency of this motor So for efficiency, we'll also see that there is another thing that has to be considered, that is the energy of the input energy. So compared to a conventional motor, this motor runs at very high efficiency. So for every unit of energy that is expended, it causes a rotation of this flagella, and therefore the energy efficiency is almost 100% for flagella. So no man-made machine has a near 100% efficiency of running. Flagella has a nearly 100% efficiency of running, 100% efficiency of converting the input energy into rotational motion. Okay, so I'll run this in. Yes.
Ma'am, we can hear but not clearly.
Huh? Yes?
We can't hear clearly. We
You
can
can't
hear,
hear
but
clearly.
yeah.
Okay. What I will do is I will stop the voice and I will start speaking so that you can hear me speak rather than the video speaking. So maybe that would help. So what you will see now is that they are going to make an animation of how exactly the flagellar motor works first. The second part of the animation would show you how the flagella is assembled on a bacteria. And I think that these two animations would give you a holistic idea, very simplistic but holistic idea of how a flagella really works. So play this. So this is the flagella, okay? And now they are going to zoom into the inside of the bacteria. Now you can see the flagella is actually rotating. So for the flagella, there are some parts of the flagella, and they are now trying to depict to you the parts of the flagella. So simplistically, you can think of flagellar parts as being three types. You would have the filament, that's like the whip-like thing that you saw rotating. Then there should be something that connects this whip to the cell body called the hook. So there is a hook that connects the filament that is like whip-like, that is moving, to the bacterial cell body. And there should be something that anchors the whole system to the bacterial cell membrane, because ultimately the whole idea of giving bacteria a flagella is to help the bacteria to move towards its food source, move away from toxic environments where it cannot survive, right? So it has to sort of adapt to its environment. So it needs to perceive certain signals and either run towards those signals or run away from signals. So the perception of those signals is done throughout the cell body by other mechanisms in the cell body. And then that has to be transmitted to the flagella. So definitely this whole thing needs to be embedded inside the cell body for it first to be anchored inside the cell body to propel the cell body, second for it to receive those signals to either move towards an attractant, move away from a repellent. So this entire cell body is, this entire flagella is then embedded in the cell body on the cytoplasmic membrane, the peptidoglycan through the peptidoglycan layer. So it's embedded, and this embedment is done by something called the basal body. So there are three parts. You have the filament, that is the whip. You have a hook that attaches this filament to the rest of the body, and then you have the basal body that connects it to the cytoplasmic membrane of the bacteria, and that also helps it to perceive things from inside the bacteria, and we will see that now. So if I go back, so, sorry. So you have the filament, you have the hook, and then now we are going into the bacteria. Okay, so here is this impressive mechanism. What do you see here? You see that this is the cell membrane of the bacteria. Then you have the peptidoglycan, and then, as I said, you can have an extra outer membrane in gram-negative bacteria. So it's all depicted here for you. And what is impressive about this is how it actually revolves. So this is a rotating flagella, and now you see if you look inside it, it's exactly a motor, right? It's exactly how engineers would be designing a motor. So you have all of these parts, wait, stop. You have all of these parts here that are all listed. You have, of course, you do not have to be told that these are rings because you can see that these are ring-like structures. You see that there are some rings that embed the whole structure inside the inner membrane. And you see there are rings that are actually present inside the cytoplasm, so present on the cytoplasmic base. You see that from inside the ring comes out something called a rod that is connected to the hook that holds the filament. And you see that there are some structures which are not moving. So the structure that is moving here, if I may start this video once more, the structure that is moving here is the motor. The structure that is not moving here, these rings are the stator. And well, let's play this. But if you have the analogy of the Tesla motor, compare it with this motor, then you know that the stator must be producing some sort of an energy to drive the motor. So for Tesla, it was electromagnetic waves, right? It was current that was producing an electromagnetic field that could drive this motor. But what is driving the motor here is neither ATP nor GTP. What is driving this motor is actually a proton motive force or an ion motive force. Now, how many of you have heard of proton motive force or ion motive force? I think for a change I'm going to call out a name. नहीं, रत्नेश एक मिनट वेट करो. सारिका, मैं आपके पास
Yes,
आती
ma'am.
हूँ. Tell us, because your PhD topic would almost be something similar to this. So what is exactly proton motive force or ion motive force?
Ma'am, I don't know exactly, but I can try. I haven't studied it.
Alright, फिर तुम wait करो. Let's hear it from Ratnesh. Yes, Ratnesh.
Ma'am, actually I think the H plus concentration outside the cell and inside the cell is different, and because of that, when H plus tries to move from one side to another, and in that process the force is the generated, and that is a proton motive force.
Okay, so yes, and proton, so thank you, Ratnesh. Thank you for this explanation. So proton motive force, uh, so you can have one of the reasons why our body systems are working is because of differences in the concentrations of ions, as Ratnesh has pointed out, between the inside of the cell and the outside of the cell. That's the reason why we have neuronal conductivity. The reason behind mitochondrial function and ATP synthesis. And the whole thing comes from the fact that membranes are not permeable to ions. Membranes do not allow ions to move. You need a way for these ions to move from one side of the membrane to the other. And that creates a very easy way for the biological systems to create differences in the ionic gradients or in the ion concentrations between one surface of the membrane versus the other surface of the membrane. In this process, what it does is it creates a small charge difference, a really small charge difference, and with that you can actually calculate some sort of voltage, that the change between the charge difference between one surface of the membrane and the other. You can also think of it as a small change, a small potential energy difference between these two faces of the membrane. What is done here by the flagella is that it uses the ion difference between the outside of the cell and the inside of the cell to its advantage to drive its motor. So outside of the cell has a very high concentration mm, of ions such as protons, whereas inside the cell these ion concentrations are less. So the stator that is associated with the flagellar motor actually is a channel that allows the passage of these positively charged ions, such as protons, but may also be sodium ions. So we'll have a look at this video once more, and this time what I would like you to do is to simply watch these stators, and you will see these small dot-like structures, white dot-like structures, which here are the ions. You see there are more dots outside than there are inside, and you see these dot-like structures passing through these stators. They will pass through these stators inside, and every time one of this ion moves, it creates a sort of conformational change in the stator that drives the motion of the motor. How it does, we will see it again in this animation, but just watch it. You see that these white ions, these white blobs are moving through the stators. You see that? These white blobs are moving through the stator, and that is the thing that is powering this entire rotor to function. So this is the rotor, this is the stator, and you see protons that are moving through the stator that is then creating a sort of conformational change for the stator in such a way that it creates rotation of the motor. So everything depends on this proton flow, but it could also be sodium ion flow. Okay, here they are specifically mentioning the proteins that is part of the stator. Not necessary for you to memorize that, these are the names of the proteins, because as I said, there are about 60 different proteins that are present in the flagella. Not possible to remember all of them. But definitely you must remember something called flagellin. It's simple and easy to remember. And what I would like you to do here is to rather look at how these two proteins called MOT A and MOT B, which forms part of the stator and also part of the ion channel that allows these protons to move through, actually moves. And this is a very simplistic but beautiful representation of how this. So they're showing you two aspartic acid residues. And again, if you mutate these residues, you would see that the motor is not able to function. Therefore, we know that these amino acid residues are important. So how do they do this? You come back to the lecture number 12 or 11, I don't remember, where we had seen that in dynein, if you start mutating certain lysine residues to threonine in the AAA plus domain, and then you start to see which of these is actually causing a stop in sliding activity of mitotubules. You can exactly point out which AAA domain or which amino acid is really important. Similarly, if you do a similar kind of experiment here, you will be able to understand which amino acid is important for the movement of the protons and the corresponding rotation of the motor. So here they are saying two aspartic acid residues are important, and you see a proton that is going to move past the channel between the MOT A and MOT B, and as the protons move through this channel, you will see there's a conformational change, and the conformational change is going to drive a power stroke. So again, you need a power stroke, and that every time there is a power stroke, you see the motor has moved, has rotated. So then when the ions are released inside, it causes another power stroke, another change in conformation, and then another motion. So this is how the flagella in the end moves. Okay, I will stop here, and we will continue a little bit more with flagella in the next class. I'll stop here for today. And I'm going to just make one announcement. So the mini project that I had spoken about, you will find it on Moodle. And the deadline for submission is 7th of March by 9 a.m. And the deadline cannot be extended. So under no circumstance can the deadline be extended. By this weekend, you will find your answer scripts back into your email ID, your IITD email ID. So please check your IITD email ID on Monday. You will find your marked answer scripts with you. And the third announcement, what is the third thing that I want to announce? One is regarding the answer script, second is regarding the mini project. I forgot. Yeah, so the mini project, your answer scripts, I cannot remember. I'm sorry. If I remember, I will maybe I'll send a mail. I can't remember the next thing. So if you have any questions regarding today's class, I'll be here for the next 2-3 minutes. You can ask me. But otherwise, we have finished the class. No questions? Okay, then we will stop with the class today. And for the mini project, if anybody needs any help, do not hesitate to contact me. I don't know if all of you are on campus, but those who are on campus can reach me out at my office. For those who are not on campus yet, you can write an email and we can discuss online if you have any issues trying to do the mini project. I have put enough explanations so that it helps, but if there is anything additional that you need, you can always reach me out. All right, students, thank you and see you again in the next class. The next class would be on, is this Saturday working day? Man, I don't
No, ma'am.
know. No? Okay, then we meet again next, I think, Wednesday. Perhaps Wednesday, Friday and Saturday. Maybe that's the order in the next week. We meet again on Wednesday. Thank you so much. Thank you.
Ma'am, in next week, there will be mid-sem break, I guess.
Oh, really? There's no class next week?
Perhaps I have
Perhaps
to
not.
check
I have
it.
to check the. I have to check my calendar. I'm not very sure. But if there is a mid-sem break, I'm not going to schedule classes. But yeah, if there is a mid-sem break, I will not schedule a class. I will schedule the class when the mid-sem break is over. I will check online, and you will be able to see it on MS Teams anyway if I schedule a class. All right. Thank you. Thank you so much, and see you in the next class then, whenever the class is. Thank you.
Thank you, ma'am.